So we have a couple slides here on the company line, and then I'm going to get into some fun stuff, sort of a virtual tour, and talk a little bit specifically about the facility. But we got to get through how the process works before you see that. So, of course, we're committed through proper animal model selection and use. As I mentioned, you have some very competent people on the review side who are saying, well, you want to study fat metabolism. Well, what comes to mind if you're studying fat metabolism in terms of animals? What types of animals have very special fat metabolism? Bears, because they hibernate? Well, if you want to do a study and your group size is 15 and you have 14 groups and you've got to have controls and you've got to repeat the whole thing, how many bears is that? Too damn many. Okay? I'm not going in to feed the bear for you. You get the younger grad student or the undergrad. So, if you submit a protocol with bears, it's not realistic. Okay? It's dangerous. And so they'll say, well, if you really want to study fat metabolism, what other animals are there? So you think smaller, okay? Think smaller. And I'm not talking about midget bears or anything like that, or baby bears. I'm talking about, there are, some of you may be studying obesity in humans, and you know that there are certain rodents who have been developed that are obese animals. There's also things that are naturally occurring, like lemmings, who have a very special type of fat metabolism, and they sort of pseudo-hibernate. And we had a very interesting colony of lemmings here on campus where the researcher was studying fat metabolism. So we help you out with that. Reduction in research variables due to disease and other causes. I'm not going to point anybody out, but earlier today I very inappropriately yelled at someone who came into our office wearing their booties, who had been wearing them inside the animal facility. And the reason that we do that is to control pathogen spread. It's not to keep the floors clean necessarily, okay? So there's big things that go on why we ask people to do certain things, and maybe our training isn't as good as it should be to tell you, why you do those things, but they are important. And if you're studying something, you want your animals to be clean, happy, and healthy so that you don't have those variables interfering with the science that you're pursuing. And of course, compliance with humane principles and legal requirements. You got to do it this way because Uncle Sam says you got to do it this way. And I'll go through a little bit about that. So understanding all of this can be a little bit tough, and I used to have a really cheesy picture of Tom Cruise up here because I was sort of allude to that it's an impossible mission to understand that. But I'm going to get away from that bad joke, and I'm going to show a couple boring pictures of books. There are a lot of rules and regulations, okay? And it's our job to make sure you know what it is you need to know, and nothing more, hopefully, unless you're just interested, about what you need to do, and to train you in specifically what you can and can't do with the animals. And so the book on the right there is the NIH Guide for the Care and Use of Lab Animals, and it's gone through, I think, 7 or 8 or 9 editions. The next one is due out next year. And it's basically the Quran or the Bible for animal researchers type thing. It's what you can and can't do. It's broken up into 4 sections like veterinary medical care, animal husbandry and housing, and protocol review and health and safety and issues like that. There are all types of standards. It says, would you have guessed that the humidity for a rodent has to be exactly between 30 and 70%? Well, you're thinking, well, that's easy. Look at that. That's a huge range. Well, those of you who have been in Alabama long enough and experienced the summertime know that 70% humidity in the summer is nice. And so we have to work very hard to lower the humidity to get it there. And that's a huge cost if you think about it. So, we have another historical section here, and this is a dog named Pepper, I think for obvious reasons. Anyone here have a Dalmatian? No Dalmatians. Okay. Well, when they're born totally white, and they develop spots, and that's a very unique pattern for them, very analogous to our fingerprints, okay? We are distinguishable by our fingerprints as the Dalmatians are by their spots. And this is an old picture back in the 50s, more or less, and Pepper, long story short, Pepper lived on a farm. Someone picked him up and sold him to a hospital to do some cardiac studies, and there was great studies, but poor Pepper was euthanized as part of the study. Now, I would be devastated if that was my dog, okay? You have to draw a line between your research animals and your pets, so to speak. Emotionally, for sure, just like a human patient would with the doc would with their human patient. So Pepper's picture was published in a very prominent magazine at the time. This is back in the 50s. And eventually it made it back to the farmer, and he saw this picture and recognized, he said, "Dadgummit, that's my Pepper." And he could tell it was Pepper because the spot pattern, okay? And so he went all the way to the hospital to find Pepper and get Pepper back, and only lo and behold find out that poor Pepper had been euthanized. Because of course the article doesn't say, you know, dog killed in study. It says great things come from research. And so he was devastated, as I'm sure any of us would be. And it caused a lot of public outcry. And people were saying, well, how could this have happened? And there was a lot of letters to the editor of Life magazine. That was kind of the preeminent magazine back in the 50s. And I've heard tell that they had more letters to Life magazine about this issue than all the letters combined about the Vietnam War. And you think about that, you know, war is obviously a very controversial subject. So it was on the public's mind, is my point. And they said, well, how come the government doesn't have any rules or regulations in place? And don't these scientists know better? Don't they have common sense? Why are they going out getting pets to be used in research? So it caused a lot of public outcry, and that's Pepper's contribution. It all came down to that one dog, so to speak. And it spawned the anti-vivisectionist movement. And this is a picture of a naked lady. And so, you know, I mean, well, I guess that's not true. She does have a sign and some paint. But she is a member of PETA, and she's inside of a cage dressed up as a tiger, and she's protesting the use of animals in zoos and circuses. And actually, they've had people like this up at 5 Points. I know you're thinking, yeah, they do that every Friday night, but, but actually, they do have their scheduled protests, and they come and they do this type of thing. And she's very sincere in what she's doing, and she's protesting, trying to make a point about it. And it's very important. I mean, I don't mean so much to poke fun at her. It's just, it's a nice picture. There's also the other approach where you just kind of come out there and you protest with your clothes on. So very important, though. They brought this to the public mind. I mean, they're sensationalists, to be sure. They want to get a lot of bang for their buck in the media. And they do protest here. Twice a year there are scheduled protests on campus. They're very small groups. They usually only garner about 5 or 10 people. And if you've not heard about it or seen it, it's because it doesn't garner much attention. And I do have a video clip of a local news story about that coming up here in a little bit. But it's very important. And here's the news clip. Let's see if we can get this one to go. It's about 2 years old. Maybe it's taking a minute to load, or maybe she's just shy. Well, you recognize the building anyway. You might recognize her. It's about a 30-second clip, and if it's not going to go, and if there's anybody out there who can guide me, it usually goes if I just click on it. But long story short with this, I guess, is they had this group called Stop Animal Exploitation Now. It's S-A-E-N, sane, or insane from my perspective, I guess. But they're just like PETA, and they came on campus and they staged a protest out in front of Volcker Hall, and it's not that building there, but Volcker Hall, and they were protesting that we had not let them in to see the animals. And they take up an enormous amount of time from us because we are a public institution and we're obliged to release records because of the Freedom of Information Act. And so be very careful what you write in your protocols. You don't want to write things that are, you know, flippant and things like that. Be very respectful of the animals in the way you write it. You don't sacrifice animals, putting them on an altar and doing this. You humanely euthanize them. That's an appropriate way to reference that. So the president of Stop Animal Exploitation Now was complaining that he couldn't even get into the buildings to talk to people, and we wouldn't give him any records or give him any tours, and they're holding up signs of animals and very compromising and out of context type pictures. And of course they weren't UAB animals, and nobody ever came to my office saying, let me in, I want to see something. And you know, so you have to take it with a grain of salt. But the point is, they're out there, they represent a very important side of the story, and they are sort of what generated a lot of public interest and caused the government to say, hey, stop, we need to have some rules and standards set aside. So you've seen these 3 R's before: replacement, reduction, and refinement. Those are in one of the slides that Professor Kincaid showed you. And I put this up here. It was 1959. Again, this is before there were any rules or regulations going on. And these two British chaps, Russell and Birch, had this book called The Principles of Humane Experimental Technique. And woven throughout the book is the concept of these 3 R's. It wasn't that they were saying this is the way you must do research. They're saying if you're a good scientist, you should consider these things. So back, even before there were any rules or regulations, scientists were saying, hey, you know, there is a right and wrong way to do things. And these guys had it pretty well defined. Replacement, using non-sentient material instead of animals. So using cell culture instead of a live animal. Using a computer instead of a live animal. Reduction, lowering the number of animals used without decreasing information content or precision. That's where you could use statistics as your friend instead of your enemy, and say, you know, well, I just need 1,000 rats to do this study. No, do a little power analysis, see if you can lower that number, okay? You have to justify these 3 R's actually in every protocol you submit, or your PI does. And refinement, which I think is the most important R, and that's decreasing pain and distress wherever possible. Now, in animals, unlike humans, we're big crybabies, okay? If something hurts us, we whine and fuss about it and take a pill and go get a little Snuggie and get all happy and comfortable. Animals, on the other hand, especially rats and mice, which are prey animals in the wild, they have evolved to hide their pain, okay? That's why as researchers, when you go to check on your mice, you have to be very, very careful in how you're looking at them, because they're going to hide whatever pain, because it's a natural thing that evolution has given them. And so they're not going to fuss about it and say, yo, knocking on the cage, I need an aspirin, okay? They just won't show it. And so you have to be aware of things that could cause pain or distress, and those are 2 very different things, and minimize it. Of course, if you're studying pain, you have to somehow cause pain, and we do have pain research going on campus, and that's a very sensitive issue. But anyway, replacement, reduction, and refinement were around long before there were any rules and regulations, and scientists were thinking about, you know, the appropriate way to do things. So, to kind of sum up this first part of the talk, increase public awareness through stories like Pepper and the anti-vivisectionist movement with PETA and SANE, groups like that. Bringing about a need for rules, regulations, and policies. All right, let's wrangle these cowboys. You just can't do whatever you want. Then you have the Uncle Sam, emergence of federal oversight agencies and accrediting organizations. And then hopefully that results in what you all are being trained to do as students, ethical and responsible conduct of animal research. That's really where we want to be. All right, there's your wake-up call. So that's Barney Fife. And yes, I'm the Barney Fife of UAB, I guess you could say. I am the animal cop. But I prefer to be seen as your friend and to help you understand what it is you need to do. Again, you guys are the customers. You're the researchers. We're here to let you know what you can and can't do and draw the line. And if you step over that once, that'll be an education session. You step over it twice, you know, that's when we put on the badge and put the gun and all that business, okay? So this is an actual reference to the Code of Federal Register, Title 9, Subchapter A, Parts 1 through 4, and amendments. You can just picture Barney going on and on about that. But there are a lot of rules and regulations out there that say what you can and can't do. All right? And we do have to hold the line on those things. And if you think of your protocol as your contract with the institution, that's really what it is. You have to do only what's written in that protocol, and only what's in that approved protocol is what corresponds to all the rules and regulations. Sometimes we do have variances, but they have to be scientifically justified. So this is my only slide to sum up this boring topic of rules and regulations, and we'll get into some fun pictures. But it's very important. The Animal Welfare Act came about by the U.S. Department of Agriculture in 1966. You're probably thinking, why is it the Department of Agriculture? It's a very large agency. They have a branch called the Animal Plant Health Inspection Service, and they administer the Animal Welfare Act. Okay? Then the Public Health Service, otherwise NIH, which is funding almost all of your stipends here and, you know, paying the bills for us to be in this nice building. They came about 20 years later and said, okay, the USDA regulations are pretty good, and those were all brought about by the animal rights activists causing a bunch of concern, but we've got 20 years experience, we've got these scientists who've been doing things ethically for 20 or so years now. Let's write our own set of standards and have it based a little bit more on performance-based standards instead of black and white, what you can and can't do type things, okay? A little bit easier to digest and read. There's also non-federal groups. Anyone know someone who works in a hospital, or maybe you've been through one? You know, every 3 years they got to go through this relicensing or accreditation project called the Joint Commission in order to maintain their license and be a hospital. We have that very same thing here. It's called an accreditation process, and it's voluntary. And the important thing here is you see we've been continuously accredited since 1971, and that's before the NIH regulations were in place. So UAB knew at that time that we want to be doing things right. Let's be an accredited institution, and we'll follow whatever regulations or requirements or guidelines that accrediting group wants. So you also know, as scientists, if you have a great idea, how do you determine it's great? You just look in the mirror and say, by golly, that's a great idea, and you look marvelous. No, you talk to your buddies in the lab, or your mentor, or someone down the hall in that discipline. It's the peer review process. They say, that's a great idea. You write a grant, it gets reviewed by your peers. You write an article, it gets reviewed by your peers. It's not published until they say okay. So my point here is the accreditation process is a peer review process. It's as if folks like veterinarians who work here or myself go to another institution, check out all their nooks and crannies and all their paperwork and all their animals, and they say, yes, you are or you are not following the rules and regulations. So that's the gold standard, is the peer review process. So, as I mentioned, I have little kids. You can have all the rules in the world, but unless you have consequences for those rules, they're not going to be worth anything. And so penalties can be very severe. You can end up in jail, and you can certainly end up in hot water. If one person does one thing wrong enough, we can lose our USDA license. We can lose our assurance with the NIH, which is our contract that says we'll follow the regulations. We can lose our accreditation. If we lose one or all three of those, God forbid, we can lose all of our research money like that, to the tune of about $1/3 of $1 billion a year coming to UAB. And I know you don't want to be that one person who does that, because you probably won't pass your qualifying exam. But we hope it never gets there, and it's our job to help you stay on the right side of the line. So one more party line: no animal will be used without a clearly defined goal, either for advancing knowledge or for the education and training of students. You just can't use animals on a whim, okay? We had somebody once who wanted to study vision in tigers at the zoo, and we thought, you know, he didn't have a grant to support that, so it hadn't been through that peer review process, and we kind of nixed that project. There are certain health and safety issues, as you can imagine. The experiment will be designed to use a minimum number of animals, and then only after alternative means of accomplishing those goals have been considered. That's the reduction and refinement and replacement. We're talking about those R's again. The animal will experience the least possible pain or discomfort. Again, to me, that's the most important thing. So who does all this? It's the Institutional Animal Care and Use Committee, and that's not a big word that I made up. It's a federally mandated committee that you have to have in order to be a research institution. And we have to develop policies based on all the rules and regulations and implement those policies and oversee them. And so, as you can imagine, it has researchers on the committee, veterinarians, non-scientists, experts in occupational health and safety, and I think the most important people are those who are required by law to represent the general public. They can have no affiliation with UAB, they can't have a spouse or an immediate family member who works for UAB, or they can't be retired from UAB, they can't have any connections. So we have a retired lawyer and the veterinarian from the Birmingham Zoo serving on our committee to represent the public. And we have 41 committee members, and the law, depending on which law you are reading, the USDA says you need to have a minimum of 3 committee members, and the NIH says you need to have a minimum of 5. Now, we didn't create a committee of 41 just to have a bureaucracy. UAB is a really big place. There's a lot of different types of research going on, so we need that variety of expertise on the committee. So we have folks from all different departments on campus. These are volunteer faculty members, along with our veterinarians and health and safety experts, okay? Lots of people. We have lots of protocols to review. During a typical month, we might process about 200 protocols. So if you're ever wondering why it's taken so daggum long to get your protocol approved, we have a lot going on. But call us. We'll try and put it to the top of the stack as much as possible. So the things we do, as I mentioned, the policies, we review and approve animal protocols. The system is set up that you can't purchase your animals until you have an approval number. At least that's the way it's supposed to work. I know the breeders out there have a loophole, so to speak. Ensure for your training and safety, investigate animal care and use issues, and suspend non-compliant activities. So in this business, the F word is the S word, okay? If you're suspended, you're basically on a list that you're not going to be able, likely not be able to do animal research for some period of time or maybe never. And that can come from a single infraction. And again, we try to help you to not get there, but it's a serious business. Yes, we do animal research. Yes, things go wrong, but we don't tolerate things staying wrong repeatedly. There's too much at stake for the animals and the institution. Okay, so just like all good plans and scientific endeavors, you know, you have your eureka moment, you got your bright idea, or more likely your mentor says, do this, and you have a plan, you submit a protocol, the research occurs, and then you get more ideas, and it's sort of a cyclical process. And there's a lot of things involved. I used to have a chart up here that I took out because it was so busy. But it shows from the point of conception of the idea to the point where the project begins, you've got to go through many, many steps, many different people and hands on those protocols before it approves. It doesn't come to my desk and I say, wow, looks good, $5 approved. Doesn't work that way. It's a very engaged committee.